Now, our next speaker is Gary Craig, who received his degrees at University of Maine at Orono, which is only, a few states in a province or so east of here. And the degree was in chemistry and chemical engineering. He received bachelor degrees in both these subjects.
And determine where the tumor will have an impact on an outcome. In our lab, we have a lot of projects. We have had some projects, whole animal imaging, using gold nanos for this, using X-ray tomography. I won't be talking a lot about that today. Basically, I'm going to be talking more of a histology view. We have some section pancreatic tissue, both healthy. Here you can see a duct, and these are all our cells. This is that's been immunohistochemically labeled for stroma. Stroma is basically the immune response to it's scar tissue. It's an immune response to anything that goes wrong in the body. Is this scar tissue yourself or when there's this is the majority of tissue in it actually. So this is a very looking piece of tissue, and this is not this is pancreatic cancer. As you can see, the features are distorted. There's a amount of stroma in the tissue, and this is that we want to take advantage of in our labeling. Now, our lab, like I said, have very many projects. We have a lot of interconnected themes. We want to understand nanoparticle photophysics. We want to develop imaging tools, and we want to develop nanoprobes. We take a biological question, such as how do we find pancreatic cancer, and we try to use the tools at our disposal to help take a measurement that will further these ends. We're engineers. If any of you saw my name on the program, I have an EIT after that. For those of you who don't know, that's engineer in training. It basically means I'm halfway to a professional engineering designation. I have to go into industry to get that for a few years, take another test. That's in the future. I hope to get there someday. But our design requirements, we want our particles to be small, nanometer sized. We want them to be shape and size tunable, non-toxic, ideally, biocompatible. This is a big one, as many people have touched on today. They're hard to buy, but they're cheap to make. And if you can figure out how to make them, synthesis is the way to go. So for our particles, we typically use either silver nitrate or gold chloride, noble metal nanoparticles. Reducing agents, sodium citrate, sodium borohydride. It's a strong reducing agent that's used in some of the more complex synthesis. Surfactants, we use CTAB, acetyl trimethylammonium bromide, to help produce the rod shapes. And polyethylene glycol, which isn't really a surfactant, but we use that for surface stabilization and biolinking of our nanoparticles. So ideally, these particles should be stable, not aggregate. Typical gold colloids are not stable in biological conditions. Changes in temperature, pH, and concentration will cause aggregation, and we don't want that. Now, here's a slide that shows why we want to use these nanoparticles, and I'm going to distill this to its essential point. Particle scattering is proportional to the 6th power of the radius of the particle. Basically, that means that our scattering of the particle is extremely nonlinear, and we can take advantage of this for our contrast mechanism. Here we have a picture of some nanoparticles. On the right, it looks like a wide side distribution. You can tell this by the particle color variation. On the left, we have a graph of three different types of particles. One looks like, in the green, we have a bimodal distribution. Larger particles scatter longer wavelengths. So we have large particles in red in a very narrow distribution, and smaller particles in blue with a wider distribution. Now, this is a really neat slide that we put together. For the nanorods, they exhibit a polarization dependence because they have 2 aspect ratios. If you look at a nanorod on its axis, it'll be small, but if you look at it on its length, it'll look like a larger particle. So we actually see these things in solution rotating and blinking different colors, going from green to red. And here's a UV vis spectra of various lengths of nanorods. So as you would expect, you see different size, different curves for different aspect ratios of particles. Now, surface modification. I mentioned that these particles, as they are not stable in biological excuse me, not stable in biological conditions. So we've been working with Dr. Wolfgang Eck, formerly of the Jackson Labs, he's now at the University of Heidelberg, to develop this linker molecule. It's a covalent linker, dithiol. There are a lot of monothials that are being used, but this one is much more stable. Basically, the dithiol makes it so it's covalently bound and it will not come apart, which is good. It also has a carboxy group on the end, which we use for our coupling chemistry. So this provides stabilization and a point of attachment for our target molecule, which is an antibody, which I'll get to in a little bit. Now, here's an interesting video that I wanted to show. This is an NIH 3T3 cell, and as you can see, it's not a happy cell because it's in deionized water. It's also been incubated with wheat germ agglutinin-coated nanoparticles. So you can see some of the particles have agglomerated because wheat germ agglutinin is a glycoprotein, and it just physiosorbs to the surface of the particle. There are also some that are floating around in the cytoplasm here. Now, like I said, this is in deionized water, and it's blowing up like a balloon. And in a couple of seconds, it explodes. So this is a neat video. It's also proof positive that we can get these particles into the cell. And it's a fun video. We have our probe system. We have polyethylene glycol to make it biologically stable. We need a targeting mechanism. Antibodies are a convenient targeting mechanism. They're used by the body to target various intruders and other things that the body doesn't want. We use monoclonal antibody F19, which is specific to tumor stroma. This was developed by the Ludwig Institute and specific to fibroblast activation protein alpha, human fibroblast activation protein alpha. And so we couple this to the polyethylene glycol moiety via NHS EDC coupling, and then we purify by size exclusion chromatography. So this is a cartoon of our conjugate. It's not to scale, but you get the basic idea. Here is the PEG dithiol synthesis route that we use. I don't need to spend a lot of time on this, but just to give you an idea, most PEGs cost on the order of about $400 a gram. This one you can't even buy. You really have to make it yourself. So once we have our particles conjugates, when we make the particles, we obviously get a size distribution of particles, and we want to purify them. On the right here, you can see we have a wide distribution, and on the left is a relatively monodisperse distribution of nanoparticles around 15 nanometers. So in order to purify our conjugates, we do size exclusion chromatography on them. We're in the process of attempting to scale this up. But this is, you can see that there are various, you get dimers of the antibody, you get unbound antibody, and this helps to purify it for use in our targeting mechanism. Here are some TEM images of the various fractions of size exclusion chromatography. You can see we have an aggregate over here, and in the second one, I can't see it, but I believe there's a dimer in one of them. There's a dimer. And so we want to remove these polymerized nanoparticles. Now, just to give you an idea of what we're looking for, these are NIH 3T3 cells that are happy. They're not in deionized water. On the left, they've all been labeled with gold, similarly to the one in the video. On the right, half of them have been labeled and half of them haven't. So this is an idea of the contrast that we're expecting from this targeting system. Now, this is the gold nanoparticle F19 conjugate labeled tissue. On the left, we have cancer with pegylated gold control, and on the right, it's cancer with a non-specific antibody bound to the nanoparticle. When we do our monoclonal antibody F19 staining, however, we see a large contrast enhancement, and we can see features similar to what we see in the immunohistochemical staining. So this is very promising. Just to show all these together, some of the things we're working on is reproducibility. We want to be able to repeat this experiment many times. We want to optimize the nanoparticle geometry. We want to be able to do lots more tissue studies. And we're also working with some collaborators at the University of Southern Maine to do some toxicology studies on various types of nanoparticles. Now, once we've labeled the tissue, the obvious answer is what we seeing actual labeling. And luckily, gold scatters electrons too. So we wanted to try to confirm our labeling. And we're still in the process of this. Our initial TEM studies were less than exciting. One of the problems we have is that we can't get a lot of the antibodies, so we're thinking about other routes, other antibodies to try. Gold nanoconjugates, in conclusion, have great potential in cancer diagnosis. The pegylated gold is extremely stable. The F19 antibody conjugates show great potential in cancer labeling. Future directions, like I said, it's hard to get F19. We're looking at a couple different antibodies. We have done some mouse studies, and so if you do a xenograft of a tumor cell into a mouse, this antibody is specific to the immune response of the tumor. So if you grow a human tumor in a mouse, you're not going to get a human immune response. You're going to get a mouse immune response. And this has been a problem. So we're looking at different antibodies. We're also, like I said, investigating other contrast mechanisms with these nanoparticles, but I'm not going to talk a lot about those today. Couple references, briefly. This is our group. My boss is the guy in back there. And one thing I need to mention is support came for this project from the Memorial Sloan Kettering Cancer Center. And I have a rare opportunity for you. We've been talking about nanobots in the cell, and would anyone like to see what we're going to look like in 50 years with trillions and trillions of nanobots coursing through our veins? So this gentleman was on 60 Minutes a couple months ago, and he has a condition called Argyria. He's been taking colloidal silver as a dietary supplement for many years in excessive amounts. And so it's actually funny because we had, on our initial mouse studies, when we put gold through tail vein injection into the mouse, our researchers called us and said, yeah, the mouse turned blue. And we're like, what? Okay. Well, this gentleman has the same condition, so this is the future of nanotechnology. Open up. And I'll open the floor to questions.
Well, Craig told me that this was, as he also told you, was his first presentation at a science conference, and he said he felt a little nervous. It didn't show, and I think it's fair to say it was a great beginning talk. And we'll look forward to the one 10 years from now when you're totally polished. Did we have some questions? One thing I just wanted to mention, a cancer of the pancreas, you pointed out, is a really bad news cancer. You know, the 5-year survival is about 7 or 8%. It's one of the deadliest of the cancers that we have to deal with. It's also a candidate for possibly being of viral origin. And that's based primarily on epidemiology, in which you, in a series of studies, you find a higher prevalence of hepatitis B in patients with cancer of pancreas. It's not as striking an association as with cancer of the liver, but it means if there's an association, that means there's some kind of pathogenic connection. So, and again, this idea of targeting rather than cancer cells with, if it turns out to be viral, cells with viruses.
It's interesting you say that, because one of the main reasons that patients with pancreatic cancer die is not from pancreatic cancer, it's from metastases in the liver after resection.
Yeah, good point. And anything further? If not, thanks again for a really interesting talk.